We're starting to go live now. Thank you everyone for joining us today. This is our regular Friday seminar in the Open Source Neuro series, which is part of the Worldwide Science Initiative. This series is all about open tools and methods in neuroscience that we collect in our website, open-neuroscience.com. You can upload projects yourself to the website and sign up to give a talk about your own projects using one of our forms. We welcome your contributions, and you can also share our content and follow us on Twitter. You will find all relevant links in the description below. Today, we have the pleasure of hosting Dr. Carsen Stringer. Carsen Stringer is a group leader at HHMI Janelia Research Campus. She did her postdoctoral work with Mary Spacitario and Karel Svoboda at Janelia, and did her PhD work with Kenneth Harris and Matteo Carandini at University College London. She develops tools for understanding high-dimensional visual computations and neural representations of behavior. We will now listen to Carsen's talk about SWIT2P, a multipurpose functional segmentation pipeline for cellular imaging. And you can ask questions directly in the YouTube live chat, or if you prefer, you can continue the discussion with the speaker while the talk is developing here in Zoom. So hello, Carsen, and thank you for being with us today. Please go ahead.
Thanks so much for having me. Can you see my mouse on the screen? Awesome. All right. So I'm going to be telling you about SWIT2P, our processing pipeline for calcium imaging data. So what SWIT2P does is it takes raw images. They can be in several different formats. We support TIFFs, H5, SBX, mesoscope TIFFs, and Bruker TIFFs. And the first thing SWIT2P does is it does motion correction. So it makes sure that you might have some movement in your imaging, because a lot of these imaging techniques are done in vivo, so the mouse or animal can move around. And so you want to first correct this motion so that you can accurately get the fluorescence of a single cell. Then the next step is detecting ROIs in these motion corrected images. And then in this detection, we have the ability now that you can use Cellpose alongside our functional segmentation algorithm if you choose. So I'll tell you a little bit about that, a new feature today. After you segment the cells, SWIT2P then extracts signals from each of these cells, basically summing the pixels in each mask on every single time frame, and that gives us these traces here. And then the output of SWIT2P can be in an MPY file, a MAT file, or now we also support Neurodata Without Borders outputs. And then and then you can load the output. If it's an MPY or NWB, you can load it into our GUI and explore the data afterwards, or manually curate it after you've segmented it. So SWIT2P also is fairly memory efficient. So it uses around 8 gigs of RAM if you run with the standard batch size of 500 frames. You can reduce that if you have a smaller computer. And it's relatively fast too. So on this example here, you have 4,500 frames, motion corrected in 9 minutes. The cell detection and extraction is also very fast. This is, for example, if you have 40,000 frames, that will run in around 9 minutes. So the first part of the pipeline is the motion correction step. And so this part, we're going to basically take this movie and try to figure out the motion and correct for it on each frame. And so we have a reference image. I'm not going to go into how we compute that, but you have this kind of crisp image that you're going to try to register every frame to. And every frame itself can be kind of noisy, like this example frame here. And we want to try to match it to this frame as well as possible. And so if you look at the difference between these two frames, you can see this red and blue offset here. This cell is offset from the reference image, which means we need to move it in order for them to align correctly. And in order to figure out this alignment, we actually whiten the images first rather than just using the raw images. And so you can see here the edges of the cells now that we've whitened this reference image and also this example frame. And now you can see with the difference, you can see these really sharp edges that we need to align compared to before when we don't whiten the image. It can be a little bit harder to figure out how far you should move things. And so this is called phase correlation. It's a cross correlation of whitened images. That's basically the definition of phase correlation. And it basically means we have our whitened reference target and we have our whitened frame, and we're going to basically check at every single kind of position. We're going to slide this frame across our target reference and see which position has the highest correlation with our frame. And so in this case, you see the cells on the right are offset. And in this case, they're offset by 4 pixels in this direction and 1 pixel in this direction. And that gives us this peak on this map. And so using this peak, we can move our frame back to where it's supposed to be. Then just for reference, if we weren't to whiten the images first, if we just did a cross correlation, this is what the peak would look like. And it's much less sharp than the peak after we've whitened the images. And whitening is a fast step, particularly in the Fourier domain. And cross correlation, to make cross correlation efficient, you also do that in the Fourier domain. So these computations are very efficient, and so it makes sense to always just whiten the images if you have to work in the Fourier domain anyway. All right, so that will correct rigid motion, so if the whole frame is moving, but you could also have segments of the frame moving in different ways, particularly if the mouse is kind of shifting its head or something like that. So you might want to divide. So what we do is we additionally have a step for non-rigid registration where we divide the field of view into these blocks, these different overlapping blocks here. And now instead of computing the phase correlation for the whole frame, we're going to compute it block by block. And so we get each of these little maps like I was showing before, but now we have it for every single block. And each of these little maps corresponds to some size of a shift in a certain direction. And that tells us how far we should shift each block. And in order to get that at the pixel level, we upsample these arrows basically and get a picture that looks like this that tells us where every single pixel should move. And then we do this, and then we shift the pixels using bilinear interpolation. And we've implemented this in Numba to make this efficient on the CPU. All right, so if we look now, here's the video I showed before, a crop of it. Now that it's been motion corrected, you can see that these cells are basically inside these circles are not moving at all. And you can kind of look across the whole frame and see that generally, for people who are used to looking at these things, you can tell if there's motion or not, but it can be harder. So actually, in general, it's hard to tell how well you've done with motion correction. So we've also come up with metrics for how to estimate how well you've done motion correction. And these are implemented in the GUI as well. So as a user, after you run Sweet2P, it automatically runs the metrics for you, and you can check if your registration worked well or if it didn't. And if it didn't work well, you might need to change the parameters of your registration, or you can open an issue on GitHub to see if we have suggestions kind of how to make things better for you. So how do we quantify motion? And so this is difficult for a couple. The main reason is because basically this frame is constantly changing because there's activity. So it's hard to tell if things are moving or if it's just activity. And so to quantify motion, we actually do this trick where we take this whole movie and we take the principal components of this movie and we see if those principal components are moving. So if the frame wasn't moving around at all, ideally the principal components of this movie should be the activity itself. Like these cells are lighting up, then these cells and so on. And so that's what the principal components should look like. But if there's motion, actually these principal components will look like shifts in space of the cells. And so we take the principal components, we have U, and then we have the V version, the time trace of this. So U is going to be our pixels by components, and V are our components, are our time by components. And so we have this trace that looks like this across time we extract from this movie, the movie in time. And so this is how the first principal component of the whole movie changes over time. We've subsampled this. This is around, usually we take around 7,000 frames per movie. And so this is what it looks like. And now the question is, does this principal component, how it's changing in time, does it correspond to motion? And so how do we answer that? So we actually look at basically the frames where this V is very high. We'll take like the top 20 to 50 of those frames, and then we'll take the bottom 20 to 50 of those frames where V is low. We'll take the averages of those, and we'll see if those top frames and those bottom frames are shifted from each other in some way, or if their difference is just activity differences. And so it kind of is hard to tell in static images. So in the GUI, actually, we have when you do this, when you open up this window, you also have the option to kind of play them side by side. So they flash back and forth, the top and the bottom. And so the hope is that, and again, this is more people who are looking at these things a lot can tell better, but if you kind of look, you can see that the flashing, it looks like cells are just turning off and on. It doesn't look like parts of the field of view are shifting. And actually, I'm sorry, I should have put an example in here where they're shifting. I'm showing an example where things work well, where we have non-rigid registration on. But if I had non-rigid registration off, you'd see, for instance, in the corners, things are kind of shifting as this thing flashes. And so we have our top and our bottom, and this is how we quantify our registration performance. We basically do this registration step I was saying. We register the top frame to the bottom frame and see how different they are from each other, how far moved from each other they are. And we see that there's basically the registration offset is very small in the rigid case and on average across non-rigid blocks. And then the non-rigid max, basically which block moved the most across all these different blocks that have been registered, was around like 0.2 pixels. This is in pixel space, so this is quite small. So SweetUP did a good job on this relatively high SNR recording on getting on doing registration. And this was a recording with a lot of non-rigid motion, but it was high at SNR. So depending on your recording, our approach may work better or worse. We also have the option in SweetUP, if you have a non-functional channel, like if you have red cells labeled in the background, you can do registration on that channel. And that might be that might work better for you than using the functional channel, particularly if you're you have low SNR recordings. Okay, so I'm going to start with cell detection now. Are there any questions here or on the YouTube before I go on?
There don't seem to be, not on YouTube, but, oh, how do you quantify SNR, says Ferry?
That's a good question. I would say it's kind of, you'll have kind of these cells lighting up, and you'll have kind of traces of these cells. And you could look at the trace of that cell versus kind of the trace of some pixels outside the cell, and kind of how much those pixels change over time. And that will kind of be like your shot noise. And then you have the pixels inside the cell and how much those are changing in time. And hopefully the amount that those pixels are changing in time is much bigger than the surrounding noise. But that's not always the case. Depending on where you're recording from in the brain, your prep, a lot of things, it might not look so clean. So there will be neuropil signals there too. So it's a little bit trickier. But yeah, you could take, for instance, random pixels around the whole image that I've actually, this is just an example. I haven't actually done this, but you could take random pixels all over the frame that aren't pixels inside the cells, and then compare those, because the neuropil will be spatially correlated. So if you take a bunch of random pixels, you might get a better sense of some things. Yeah, so fnew.mpy is the neuropil trace. Yeah, so you could look at that. Someone also asked if after registration there is a bit of motion but a lot of change in activity, can the PC metric fail to identify it? So that's why actually we take several PCs. So hopefully there will be, you might have the case where the top PCs will be activity, and then the later ones will be motion. So yeah, it really depends on kind of the recording. But if there is some motion, it's of a decent size, if it's across enough frames, it should show up in the top PCs. If it's only a few frames where it really messes up, then it might be harder to get it with this kind of method. Okay, so now going on to the cell detection. So we have basically, this is the video I showed before. So we have this XY time movie of our neural activity. You can, for instance, take the maximum value of each pixel across time, and that makes this nice max projection image. And so one thing you could do in order to detect cells is just to do it in an anatomical fashion, where you basically just work with a single image rather than using functional information across time about how cells are firing. And so if we do this, we have this now implemented in SweetUP. You can use Cellpose in order to find these cells. In this case, in this recording, we found around 679 cells with Cellpose. In this recording, we actually would find more cells with Sweet2P, and I'll show that in a second with the functional segmentation algorithm. But in some cases, it can be helpful to run Cellpose, especially maybe if you're tracking cells across days and you know you have inactive cells, you'll be able to detect those cells maybe in the mean image, but you might not see them using functional segmentation because they didn't actually have any activity for the algorithm to use. And so here I'm just showing in the options menu in Sweet2P, you can turn on this anatomical only detection. And some examples where it might look nice is for instance, in hippocampal recordings, you might get nice segmentation with Cellpose that might, in some cases, rival the functional segmentation. And then also kind of, this is a deeper brain recording where you also might have an advantage of using Cellpose over a functional approach where you're in maybe a less high SNR regime. Okay, so how does but in a lot of cases, the functional segmentation will be better and you'll find a lot more ROIs that way. You'll find a lot more cells or whatever kind of region of interest you're looking for that way. So how does the functional detection work in Sweet2P? So we take our movie, and this is very big for lots of people. This could be hundreds of gigabytes. And now we want to try to find cells in this large movie. So the first thing we do to kind of reduce it to a manageable size and also kind of to reduce redundant information is we bin in time at the same timescale as the GCamp timescale. So for instance, if you're imaging at 30 Hertz and you're using GCamp 6S, so you have a timescale of around 2 seconds, we'll bin the movie at 60 frames. And so we'll have this bin movie, which I'm showing here, which still you can see a lot of activity in it, but we've kind of been able to reduce the size quite a bit without hopefully losing too much information because of the long timescales of GCamp. And so now we take this bin movie and we're going to try to find the cells here. So we're going to high pass filter in space and time. Oh, I see a question that's about Cellpose. So the question is, why in a video where we have a lot of cells, like your hippocampal example, would using Cellpose work better? So it depends on your definition of better. So I think I'll go through kind of how the functional segmentation works. So it's finding cells that are active. That's the difference. But then if you use an anatomical algorithm, it's going to find cells whether or not they're active, but depending on if you can see them in the mean image. And so maybe if you have a really, really clear expression here that's relatively high, a high baseline expression, and you can see the cells, you might find cells that a functional segmentation wouldn't find because there are cells that you found that are inactive. It's a question of whether or not you want those cells. Like ultimately, you might not use those cells for analysis, but maybe you want them if you're coming back day after day to the same population and you want to know, oh, these cells were inactive here or they're active on this day. And then you can maybe track them more easily than if you're just using a purely functional algorithm. But it definitely depends on the question and the expression and those sorts of things. I generally would recommend using the functional algorithm if you can, because you're going to get better results and more. The ROIs are going to be determined by the functional activity of that cell. So it'll be a better ROI because it really cares about the correlations between pixels. So it'll be more well-defined than just using anatomical information. Okay, good question. All right, so we're going to try to find cells in this movie. And to do that, first we're going to high pass filter in space and time. And so high pass filtering in space is going to really help us remove, in particular, the neuropil, which are these kind of slowly varying signals that are spatially localized. Oh, we have another question about temporal binning. So this was the last slide. So would this temporal binning predict that you'd find the same cells using a faster GCamp tau parameter, even if you are using a slower GCamp? To find the same cells using. Yeah, so you could use a smaller tau and you should still find the same cells. You just you're basically going to be processing something bigger, a bigger movie. It might be a bit slower. It might not necessarily fit in memory as well as if you bin more. But in theory, you should find the same cells regardless of the binning, as long as you don't start binning too much, and then you start losing information because you're binning bigger than the timescale, this kind of slow timescale of GCamp. If you bin too much, then you are going to start missing events that you wouldn't otherwise if you don't bin too much. Another question is, would Cellpose work on brain slices from histology? And that depends on the brain slice and the type of cells. So Cellpose has been trained on images like calcium imaging images. So if your cells look kind of like that, it will work well. If you have really small cells, like little dot kinds of cells, I recommend there's a tool called CellFinder, and there's other tools like this that might work better for those kinds of histology slices that are quite zoomed out. Okay. So we high-pass filter in space and time, and that gives us, now you can kind of see these cells really lighting up in this bin movie. And now we're going to try to find these regions where you can see these cells, these pixels lighting up together. So that's what we're going to call our kind of candidate cells. And so to find these regions which are lighting up together, we're going to downsample it. So we're going to be summing. And so if pixels are correlated to each other and we start summing in space, we're downsampling in space, then they're going to basically add up. And if they're uncorrelated, then you're just going to get 0. And so we downsample, we just do sequential downsampling so that we get maps at multiple sizes of correlated activity. So like, for instance, if we're just downsampling by 3, we'll be able to get kind of peaks in this map of things that are kind of small, but we'll also get peaks from bigger things. But those will be even bigger if we go down to the lower downsampled maps all the way at the bottom. So we downsample again and downsample again and downsample again. And then we're basically going to be looking for peaks in these downsampled masks, or in these downsampled images now, which we call maps. And this allows us to kind of figure out these regions where we think there are cells that we should find. And this is just an example image of combining these maps. We don't actually use this combined map to find cells, but just this is what shows up in the Sweet2P GUI as your correlation map, which is a combination of these maps. But in reality, what we do is we actually look for peaks in these maps directly. We try to find the maximum peaks in these maps, and that's what we use to seed our putative cells. And so I'll show you, once we seed our cells from these maps, we get basically here we've seeded a cell. This cell was found on a map maybe of this size. So we downsample by a factor of 3. And if the cell has the biggest, if this putative ROI has the biggest peak here, then we think it's a size of a 3 by 3. If it has the biggest peak here, we've downsampled again, and it's a size of 6. So if it has the biggest peak here, we think it's a 6 by 6. And then we downsample again, and then maybe it's a 12 by 12. And so then if we find the peak is biggest on this map at that position, we're going to seed, for instance, if it's a 12 by 12, we might seed it by 12 by 12 pixels. And so that's where we start with our cell. So this is an iterative algorithm. We take the biggest peak where we think like the brightest cell is, and then we go one by one and keep trying to find cells and kind of subtracting them off from our movie, and then just trying to explain as much variance as possible from the movie, from kind of getting these cells in this iterative fashion. It's a greedy algorithm that's to try to find these cells in a sparse way. So basically we have our patch. We are starting with this 12 by 12 that we found the peak was in that map. So we're going to start with a 12 by 12, and we're going to see what kind of activity this little square has across time. And this is what the activity looks like across time. And we set a threshold, which actually the user can change this threshold, which basically says which frames should we focus on, which are big enough kind of deviations from the average activity that will correspond to kind of actual events rather than just kind of like what might look like noise in the outside. And so we'll focus on these frames, and we'll basically say, what does this mask now look like? Which pixels basically are also active on these frames, like these pixels are, and then whichever pixels are active, we kind of add them to the map. And so we start adding them, we recompute this trace, we re-threshold, and we say, okay, these pixels are active. Are there any other surrounding pixels we can add to this map, or erase as well? You can change the size, it can get bigger or smaller. And then we get kind of something that looks like a cell like this. And we can do this, there's other cells that might look a little different. And this is how Sweet2P is able to get cells that are kind of unconventional looking, that have dendrites and have longer extensions, rather than kind of, because we're not trying to impose too much, imposing an exact shape on these things. We just want whatever, we basically just start with the seed, and then whatever shape comes out is what we end up using in the end. And here's an example of another cell with some kind of dendritic processes coming out. And so in the end, if we use this on the same recording I showed before where we use Sweet2P, we find around twice as many cells, around 1,400 cells compared to around 700 cells. So we're able to find a lot more cells than if just the anatomical approach. And so generally, there are cells that don't necessarily show up in the mean image that do have activity. And so generally, it is a good idea to use the functional approach, particularly for cortical recordings. And here's an example of Purkinje cell dendrites in the cerebellum, just to show that this algorithm can find things that are kind of elongated. This algorithm can find things that aren't conventional circular shapes, even though we are seeding everything with those little squares. Okay, are there any questions before I move on to the next part? Not on, okay, cool. So now here's a new addition from spring, maybe January 2021. We added PCA denoising for cell detection. Oh, there is a question now. Okay, so can you find processes / axons with Sweet2P separate from cell bodies? Yeah, so it depends on what you mean by separate from cell bodies. So we have used it in Bhutan recordings where, like, for instance, we've injected in thalamus and we're looking at axons in cortex, and it definitely does work for that. If you mean like dendrites separate from cell bodies, kind of the thing is if those pixels are correlated to the somatic activity, they're going to be included in the ROI. But you will have instances of dendrites that you will find with Sweet2P, and I might show some more a bit later when I'm talking about the classifier, that are separate from a soma. And that's just because they weren't actually correlated to a soma that Sweet2P could find in that plane. And so in that case, they will be separate. But there's no preference either way. It's just if the pixels are correlated at some threshold with the seed, then they're going to get included in, and as the cell is growing. Good question. Okay, so yeah, and also I think you can do lots of, you can do post hoc processing and say, like, maybe I want to focus on these different things separately, but that kind of depends on the user too. And later on, I'll show how you can cut off dendrites if you want to. There's a question from YouTube. In cell detection, do you have any annotations on the cell, like cell 1, cell 2, etc., later to find the same cell info for longitudinal images? Is cell detection manual, semi-manual, or fully automatic? All right, so good. All right, so the first question is, yeah, so we have annotations on the cell. So we return for the user kind of the image, which would be kind of these masks, that this pixels, that we have like a list of masks, that this mask is these pixels, this mask is this one, and so on. But we don't have anything native in Sweet2P. There are GUIs that are built outside of Sweet2P that we have linked on our website that allow you to match across days and to be able to track those cells. But that's currently not in Sweet2P natively inside the algorithm or inside the code. But it's definitely doable. And so then we said, the question is cell detection manual, semi-manual, or fully automatic? So it is fully automatic, but afterwards, if you want to, in the GUI, I don't have this included, but actually you can open up an image like this and start putting ROIs on the image. And you can actually draw little ROIs and click a button to extract ROIs there. And Sweet2P can allow you to manually add ROIs if you want to. In our case, with our cortical recordings that we're doing, we get lots of neurons. We get 60 to 70,000 cells per recording because we're really lucky to have this great microscope. And so we don't want any more cells. We're not going to go in and manually try to get, like, even if maybe there's a couple of cells that Sweet2P missed, it's fine. But in some cases, users might want to do that, and that's totally possible in the GUI. Okay, so there's a question. Can you speak of the lack of demixing in Sweet2P compared to CNMF and how that manifests with looking at this population data? That's a really good question. So first off, so Sweet2P lacks demixing, but it has a more complex neuropil model than CNMF. So CNMF often only has one kind of a one-dimensional neuropil model, which can be which means that you might not be doing the best job of correcting the neuropil for a cell. And so this, depending on what you might find is in CNMF, the cells are more correlated to each other because there's more neuropil signal inside of them than they would be if you subtract off the neuropil the way we do, which is basically we take an annulus around the cell and use all those pixels, and that's our neuropil trace, rather than using a couple of neuropil traces for the whole field of view. And so I think that's an important step that CNMF lacks, that we have, which kind of is, it does definitely can change your interpretations of population data. There is something that SweetUP lacks that CNMF has that can also change things, which is this demixing at a single pixel level. So basically, when we find cells, we're looking for these cells in these maps, and we can come back. We find a cell that's here like this, and it has this certain activity. We can come back and find another cell that's actually overlapping with that cell that also that has activity that after we've subtracted out the cell, there's still activity in a different subspace that's going on. So we can get overlapping masks like in CNMF. And the advantage with CNMF is they're going to actually try to, they have some model to try to kind of demix those traces from each other, that they're supposed to be more orthogonal to each other. We don't have that in SweetUP. What we do at the moment is we basically, any overlapping pixels between two cells, we discard them. And that's an option, you can turn that off. But by default, we discard those pixels and we'll only use the pixels kind of in like a crescent that are not overlapping and use those to compute the trace. So we don't have to kind of, so we don't have any assumptions kind of about what type of demixing happens. But this is definitely something we're thinking about adding in SweetUP in order to account for these overlaps. But our algorithm itself doesn't have kind of any assumptions about how these kinds of signals should interact. We're basically just trying to find these peaks in the maps. And then post hoc, I think this approach of doing this demixing post hoc might work fairly well, but we haven't actually implemented that yet. But yeah, it's a good question. All right, and then, oh yeah, there's a question. How can you find neuropil if cells are very dense with almost no background? So it depends on the case. So maybe in some cases like the hippocampus, that will be really hard. But what we've actually implemented, which I don't have in these slides, but if you look at the updates for version 10, which I had included in the tweet, there are slides about one of our updates, which includes the exact problem. If the neuropil is too dense, we have this new parameter where basically, I don't know if I have the best image to show, but you kind of, if you look at this cell here, you can see there's pixels that are bright and pixels that are dimmer. And basically, we would say that pixels that don't explain so much variance of a cell, we're just going to kind of cut those off and allow those to be used in the neuropil computation because they're just kind of relatively, they're not contributing too much to the cell activity. And so there's this parameter now called lamb percentile that you can set to try to get more pixels to create your neuropil, which you might not have been able to get before because it was very dense. But again, that depends on the recording. So this seems to work pretty well for cortical recordings where things are pretty dense, but I'm not sure for other recordings, it might be a bit more complicated. There's another question. Okay, is there a case or any case where two cells can be selected as if one cell due to them correlating closely with each other? Yes, that could happen if they were very correlated. And I think this happens less with the latest version of SweetUP, our sparse mode algorithm. But you could try to look and see basically if the activity in those two traces really is different by kind of drawing masks there and seeing. We don't have an option currently to split cells in the GUI. We have an option to merge actually, and to do some automated merges for people working with dendrites and axons. But we don't have that. Unfortunately, splitting is a bit harder, so we don't have that option. But yeah, it can be a problem, particularly if you don't have a lot of time points. So always a functional algorithm works better the longer you can record. So people will come to me and ask me like, or tell me that they turn off their microscope in between trials of imaging. Like the mouse does a trial, I turn off the microscope and then I turn it on again. But if you can, just record as much as you can and you can get as much activity as possible from those neurons. And that's what I recommend to people, because then you might have enough information to be able to demix these cells well and have robust segmentation of your cells. Or worst case, you might have to use an anatomical algorithm like Cellpose for your data. Okay. And thanks, Andre, for posting the link. Yeah, if you go to that link, there's a link to some Google Slides with more information on some of these updates. Yeah, these are all good questions, though. So thanks for asking them. And I think we're doing pretty good on time. So, all right, so this is one of the new things in the latest update, which is where we are doing some denoising to help us find cells better. And there's been some recent approaches for this, and you can check out these papers I mentioned here. But we kind of implemented the simplest thing we could think of, which is this PCA denoising, where basically we have our binned movie frame I told you about before, and we're going to divide this movie frame into patches, and we're going to take the top 32 PCs of each patch and use the reconstruction, a low dimensional reconstruction of each of those patches, rather than using the full thing, which however many pixels you have, maybe you have like, really you have 600 PCs, but we'll keep the top 32. And that gives you a frame that looks like this now, which is a much cleaner single frame than this bin movie frame. And so what this actually allows us to do, so here I'll compare, this is a relatively zoomed out recording. The cells are only around 6 to 8 pixels across, but you can see if I don't do PCA denoising versus if I do PCA denoising, I get a lot cleaner looking kind of ROIs. And actually, if we zoom in, you might be able to see a little better. We're kind of getting things that the ROIs are more filled out, kind of. We're getting pixels that are correlated to each other, that should be correlated to each other, that maybe we weren't getting so well before doing the denoising step. And in this case here, that was a transgenic mouse. So it does help, but you don't see huge dendrites in transgenic mice. But if you do this with an injection of GCAM, you'll get these really nice dendritic, you'll really be able to follow the dendrites now with this denoising step, whereas before this cell's pixels would be cut off and it wouldn't follow this long dendrite. And so I was pretty excited about this, that it seemed like it was a good idea to do this denoising, but it ended up actually breaking our somatic classifier in SweetTP. So for people who've used SweetTP, you might know, so after you run SweetTP, if you're interested in just getting somatic like basically somatic signals rather than like dendrites and other processes, we have a classifier. But this classifier can fail if cells have really long dendrites. It doesn't understand that a cell with a dendrite is still a cell. And so that's actually what happens in the denoise case. Here, I'll show some examples here. You have, this is a zoom in of the picture on the right, where these are cells that were thrown out that it doesn't think are cells. But if we zoom in on that, you can see these really nice looking cells that just have really long dendrites. And so we have what the classifier is using is it uses the skewness of the ROI. So basically kind of how many like kind of big peaks the ROI, the signals have, which is generally a sign that they are high SNR, that they're outside of kind of the baseline kind of fluctuations of the fluorescence in or the noise in the imaging. And then we also look at the compactness of the ROI, and that's the problem here, which is why these cells are thrown out, which is basically compactness is saying how far away are these pixels from the center compared to if all these pixels were just in a disk around the center point of the cell. And so if it's a perfect circle, the compactness is 1. And then if it's, I guess maybe we define it opposite, but basically if the pixels are kind of dendrites, we get larger compactness levels based on, that's what comes out of SweetTP. That's how we define it. And those end up getting thrown out by the classifier. And so I'll show you how we kind of fix this in a simple way, which is basically to just look at how basically we take the median pixel in the cell and we start making circles around, growing out circles around the cell and looking at how the area in the ROI changes as you start growing outwards. So if I start growing outwards, I start increasing in area and then it suddenly stops. So I keep drawing the circle. And what that means is basically that stopping point is around here, because now if I keep growing a circle around and see how much area I'm adding to my ROI, there's not very much because I just have these little dendrites I'm adding. There's not a lot of pixels there. And so basically now what the algorithm does is for compactness measures, it cuts off these dendrites. And now you're able to actually, and also for the aspect ratio of these cells, you're able to just use the center area and it can help the classifier a lot. And so there's still some cells that aren't perfect. but we're able to get more of these cells on the left now with these dendrites. You can see here kind of with these processes sticking out that were actually detected. This is the next part is very much work in progress that I'll tell you about. But are there any questions before I go into that? We're in that direction. Is a neuropil signal above the baseline an indication that a cell has low SNR? Yes. So if it could also be like it can depend on where you're imaging. If you're imaging like an unconventional area, maybe you have cells that like have a lot of negative going traces that are like inhibited, and maybe you'll have something weird there. But generally speaking, if the neuropil is on the same level of the cell or like slightly above, and the cell doesn't have any traces that go above that neuropil, it's generally a sign that that cell is not a great cell. And so in the classifiers for SWEET2P, we use the skewness after neuropil subtraction. So if you've subtracted off the neuropil and there's not much left going on in terms of fluctuations, then this cell will generally get thrown out by the classifier. And as a human, that's a cell I would throw out too, because it doesn't look like it has activity outside of the kind of baseline of activity in that area of the imaging area you're looking at. Okay. So this is okay, but it's not great. And so we wanted to make the classifier even better. And we tried to do that using a sort of ground truth data set that we collected. And so we have these animals where we injected this virus where GCAMP6S is tied to TD tomato. And so every cell that has GCAMP also has to express TD tomato. And so we have these kinds of, you can see the pink, I did it in pink and green, and you can see lots of these cells. So every cell that has activity should have a pink circle. There might be pink circles without activity, maybe because the GCAMP didn't express so well, but it means that we should be able to find, we kind of have a ground truth now that every cell that has activity should have a kind of this pink blob in the middle. And so what we did was we recorded 3 planes. We determined the ground truth masks in these planes and then trained the classifier on 2 of these planes. And then we tested the performance on the 3rd plane, which I'll show you. And so to determine the ground truth mask, this is what the pink channel looks like if I take away the green channel, and it's very dense. There's lots of cells that are expressing the TD tomato. And actually, fun fact, this is the first thing I was trying to segment before we started building Cellpose, and it wasn't working so well. And we were like, oh, well, maybe we actually need something better to segment these cells. But anyway, so now using Cellpose, I'm pretty happy with the segmentation we can get, and we can finally kind of start to work with this data. And so we have the segmentation here. And then, like I was saying before, there might be cells here that there might be nuclei labeled here that don't have expression. And so I'm going to basically filter out the ROIs that didn't have any GCAMP highly skewed traces and kind of recreated these masks, basically using the nuclei as seeds and saying which ones look like they have activity, which ones don't. And then this is what the ground truth masks look like. Sorry. And then we can say this is an overlay of what SWEET2P found from this plane. And so any of the yellow means that we matched it pretty well. And then if there's things that kind of aren't matched or aren't good from SWEET2P, then those are the ones that we're going to tell the classifier, those are no good. Those aren't somatic signals. We should throw those out. And so that's what that's the information we train the classifier on. And now if we look at our cells versus not cells, it looks like we're getting most of the cell bodies here, and a lot of the non kind of things like I was talking about before, but I didn't have a picture. You can see dendrites here, and they have somas probably, but the somas aren't in plane. And so if you just care about looking at somatic signals, you would want to throw these things out and just keep things here that are actually circular, where the planes where the soma that are actually in the plane you're imaging. So this is very much a work in progress, but it's encouraging that we might be able to train the classifier even better. Not our old classifier was trained on a person labeling cells, but now we can actually train on some kind of ground truth, and that might make our classifier even better. And finally, for people who don't know, you can run Sweet2P inside the GUI. There's this file menu up here, and you can click run and you can choose your settings there. And you can also then explore your data in the GUI, click on cells, merge things if you want. Also, there's manual labeling if you need it, if you think Sweet2P missed something. And yeah, and so in summary, our registration, I will say for our high SNR for sure, and in general is very accurate. And I've gone over also how cell detection works in Sweet2P. I showed how Cellpose can help in some cases, that PCA denoising might help in some cases too. And that's an option you can turn on as a user now. And then also we have this new ground truth classifier that I'm still working on, but that will be included in Sweet2P in future releases. And so I'd like to thank, so my main collaborator on this project is Marius Pakitsariou. And then we've also had two software engineers who worked with us last summer, helping us with the code refactoring and testing and stuff like that. So thanks to Nick Del Grosso and Chris Key for that. So yeah, thanks everyone for your time.
Thank you so much. That was a brilliant talk. And you were able to show us with every step of the way. That was brilliant. Thank you. So if anyone hasn't had the chance to pop into the Zoom meeting and you want to discuss further, please do so now. You can also keep posting questions on the YouTube chat. And we have one actually just now, if you can take it, Carsen.
Yeah, so sorry, you want me to read it?
As you wish.
Sorry.
I go ahead. Can Sweet2P output non-ROI pixel values in relation to my SNR question? That's to say, not SOMA, but neuropil.
I don't know if I fully so you want, so Sweet2P will now, I don't, so maybe this will answer your question. So we now have this logical that comes out of the stat file for people who are familiar with the Sweet2P output that will tell you which pixels I cropped when I did my SOMA crop. Are those the pixels you're looking for? Pixels that are not in F or F new. It depends. That F new, it depends on how dense your recording is, but a lot of the pixels are going to be in F new already. But you can make those, you can remake those maps yourself with the create neuropil masks function. And you should put an issue on Sweet2P, and I can help you with that. We haven't been saving the neuropil pixels anymore in the output because they're really big. So that's just kind of one downside. But we could add it as an option if, as a user, if you wanted to save those pixels and then ultimately find those. But if the recording is relatively dense, there won't be many pixels left in the background. If that answers your question. Oh, re-neuropil correction. What are your thoughts on the magical 0.7 coefficient versus trying to estimate the coefficient or other more source separation style approaches? Yeah, so that's a really good question. Yeah, so you can make those kinds of scatter plots to see kind of where that kind of correction coefficient should fall of F versus F new. And yeah, so we haven't had much success tailoring it cell by cell. Like a lot of the fitting procedures that we've tried seem to kind of fail, particularly like in low SNR cases. But yeah, it can be a problem. Like if you're really worried your cell is kind of out of plane, you might really need a higher neuropil coefficient. But if your cells end up in the end being relatively uncorrelated to each other, like we might expect in cortex, then you can hope that your neuropil correction was decent, if that helps. I'd be very happy if people are working on things to do a better job of neuropil correction, but I don't think it's a very popular area. I think people are much more interested in spike detection at the moment and other things like that. Okay, so I'll go on to the next question. So are people using Sweet2P with other indicators? So yeah, so I have seen people use it with voltage. but I don't think it's super popular yet. But the principles still hold the same way. Like you want to find pixels that are correlated that across time that you can find. You could also use Cellpose on some of those recordings, particularly if they have high baselines. So calcium imaging, generally the cells don't have high baseline fluorescence. So having a functional segmenter is really important. You really want to find correlated things because you don't see them unless they're firing. But for some indicators, like a voltage indicator where the baseline is relatively high, you might just want to do an anatomical segmentation. And so something like Cellpose might be good, or people have also, I think, been developing other types of techniques like that. So another question from YouTube is, can you describe how the sparse ROI detection works? Yeah, so the idea is, so it's a greedy algorithm. So we are ultimately, okay, so we have this movie here. We're ultimately trying to reconstruct this movie. Like, this is a matrix decomposition problem. We ultimately want to find masks and then basically have their activity. And if I multiply these masks and their activity together, I should reconstruct this matrix in time and space. But we end up trying to solve this problem in a greedy way, where we're trying to find the biggest cell kind of with the most correlated activity that explains the most variance. We subtract that off from the movie, and then we go in and we find the next thing with the most variance, and we subtract that off. So that's where we make these maps here, basically, where we downsample. We have our bin movie that we downsample to kind of, I don't know if I'm explaining this the best way, but like, if you think about, if you have pixels here that are kind of correlated to each other, if you sum up this square, it'll still have a high dot there in a max across time. But if I sum up a square of pixels that are just noise here, it'll just be 0. And so you kind of start growing out how much you downsample. You can see if, like, you can get things that are correlated that are of certain sizes, and that's kind of how this downsampling step works. But maybe I should have another animation here. I'm sorry, it's not super clear. But yeah, basically then we start choosing peaks on these maps. We start with the biggest ones. And then basically after we have those peaks, that's where we do this growing procedure, where we start to create the ROIs. And once we create the ROIs, we subtract those ROIs off from our downsampled maps. And so, because we've explained that variance now, and then we can come back in and find new cells. That's also why we can find cells that are overlapping, because there might be activity in 2 different directions, kind of cells doing different things. So we just subtract off that variance and then come back in, and we can find new cells. I guess that was from YouTube, so I won't get a response if that was clear. But okay. So another question from YouTube: can we get the traces from silent cells too, or Sweet2P only catches the active ones? I'm asking like this because if you do neural inhibition studies, one would like to show these traces as well. Yeah, so that's a good question. So that is where, and that's where kind of you might want to turn on this anatomical segmentation in Sweet2P rather than using the functional segmentation. That's really the only way to get these silent cells. And yeah, so that's what I would recommend. You could also run Sweet2P twice and merge the outputs of the 2 of them if you really needed, if you were missing cells in one case. Okay, and then so we have another question. I'm wondering what faster than real time means. So yeah, it basically means we run an experiment that might take 3 hours, but then the Sweet2P processing takes like 2 hours after that. It's not online processing, unfortunately, but it's basically the hope kind of with us in our experiments is you do a recording that's a few hundred gigs, you process that recording, and then a few hours later you can say, oh, I want to change my experimental setup. Like, we really want people to be in the loop with the analysis, and that's our goal with Sweet2P, is to make it easy to run it quickly so that you can come back and change your stimuli or your behavior depending on what kinds of signals you see.
Yeah, that makes total sense. That's great. Are there any more questions? There don't seem to be any on YouTube, and people here are not asking anything, right? Oh, one more, yes. Have you tested Sweet2P concatenating TIFFs from multiple days?
Yes, and in some cases when people are really, so I should say I didn't talk about this at all, but I highly recommend people who can use it. There's a software called ScanImage, and that's for acquiring your data. And so that's what we use. It's really great software. And they have the ability for you to align across days. You can realign to a previous recording you did to make sure you're really in the same place day after day. They also have a module, which is amazing. I didn't talk anything about this at all, but you have to also be worried that your plane might be moving up and down in Z. Didn't talk about that at all. But they also have a module that allows you to track in Z. That actually, we added that module to ScanImage because we were worried about that. So that's really important to us to make sure we get the things across Z. So that if you are doing those types of things where you're really careful about making sure you're in the same place, people have run Sweet2P across dozens of recordings across dozens of days, and that can work really well. It can also require a lot of RAM. So the person I know who did it used a lot of compute at the institute he was at. But that can be an option to run across multiple days. But you have to be careful that the alignment is good, that you don't need like a very big non-rigid shift between days to be able to match the cells across days. If there are really big non-rigid shifts, you probably will want to run each day separately and then post hoc combine traces, match cells basically.
So there's time for any last questions, if anyone else comes up with something. But other than that, thank you so much. Actually, we're going to be hosting Marius Pakitari next week, so you are welcome to come join us next time for learning about kilosorts. And thank you all for your great questions. I mean, I think this discussion was enriching for someone that's a developer of the tools, as we were hoping this series could be. And we'll welcome you next week. Thank you.
Thanks, everybody, and thanks